So we're gonna continue the conversation about gene expression, and before we move on to translation, so we talked about transcription just before that, and we focused on prokaryotic. In eukaryotes, there is one more step after the RNA is synthesized. This step is called splicing. The reason for splicing to occur is that the transcripts that are made in the process of transcription, they aren't really ready for protein synthesis. The reason for that is that, remember we talked about junk DNA. The reason is that each transcript contains not only meaningful parts, not only parts that actually encode proteins, but also fragments that are junk, fragments that are meaningless, fragments that are rubbish. Parts of the pre-mRNA that are meaningful are called exons, right? Fragments that are essentially junk are called introns. So introns are non-coding, exons are coding. In order to produce a correct protein, introns have to be removed. And that removal is called splicing. As simple as that. Do we understand that? Okay, it's really, that's all it is. So pretty much you have a system of proteins that form so-called spliceosome. Spliceosome, in very basic terms, it comes over and cuts out introns and stitches exons back. Right? So prokaryotes, like archaeobacteria, they do not have a process of splicing. Viruses actually can do it. Some viruses have splicing, but prokaryotes don't. In prokaryotes, mRNA that is produced as the result of transcription is ready to go. Now, one interesting feature of splicing is that if you have a gene which has 5 exons, this gene can undergo so-called alternative splicing, which is illustrated in this picture. So you have exons number 1 through 5. If you combine exons 1 through 4, it's going to be one protein. If you combine all 5, it's going to be different protein. It's going to, if you throw out 3, again, another molecule. If you throw out 4, another molecule. Does that make sense? It's like Lego. And in many cases, in eukaryotes, many genes, when they are transcribed, they pre-mRNA undergoes alternative splicing. What do you think will determine which version of the protein will be produced? So the gene can produce 3 or 4 different versions of the protein with different functions, slightly different functions. What will determine which one will produce? It's a simple question, much simpler than you think. You know the answer to it. You can go this deep, or which protein will be produced, which function. Function means what? Biological term. Function is manifested in the organism as proteins, function of the protein, is that a trait? So function is the part of what? All the traits show up as phenotype. Function is the part of the phenotype. So what determines which functions the organism is going to have? Gene, well, expression of genes. And what determines the expression of genes? The environment. Does that make sense? Does it? Changing environment may change which product of splicing will be produced. If you would, I'm not asking you to just go back and re-listen to all what we have discussed in the class, but you may have noticed that there is a pattern, repetitive pattern that we come back to always. We always talk about adaptation, we always talk about evolution, we always talk about environmental changes, expression of genes, and resulting changes in phenotype that allow organisms to adapt. I told you those concepts are going to be the cornerstones of this course. We always are going to come back to phenotype, to gene expression, to evolution, to adaptation, to explain why certain things happen. That's the major driving force in biology. Does that make sense? So splicing, again, is in eukaryotes, not prokaryotes. So the next big step is the translation of RNA, synthesis of the protein. And translation is synthesis of the protein, happens on the ribosomes, right? In fact, it's not one ribosome that pulls RNA molecule through and produces the protein. It's many ribosomes sitting on the RNA molecule like beads on the thread. And each ribosome produces the, that's the mistake on the picture, protein molecule. Does that make sense? That make sense? Imagine that you have to make copies of the book by writing it or by typing it in. Okay? You can see it and do it by yourself, or you can recruit 15 people and you copy the first page, give it to the next person, copy second, next person copies first. Okay, does that make sense? So you have like 15 people copying the same book at the same time. The book is slowly moving through the chain of people. Same happens here. mRNA is slowly moving through the chain of polyribosomes. The structure of the cell in prokaryotes and eukaryotes determines the fundamental difference between the translation in those two different types of organisms. In prokaryotic cells, process of transcription and process of translation are continuous. So here, this picture illustrates the two processes that happen almost simultaneously in prokaryotes. Let's work our way through this picture. The blue line in the image is the DNA. Okay? We're good? Certain gene on the DNA, say it's a gene, okay? It is being transcribed by the molecules of RNA polymerase. For some reason, Illustrator made them light beige, so you cannot see them in the picture to save your life. But trust me, there are RNA polymerase molecules here. Does that make sense? RNA polymerase. Which molecule does it produce? Which molecule does it synthesize? RNA polymerase. RNA, yep. So you can see the red or purple, whatever you want to call them, molecules of RNA that extend from the blue RNA polymerases. Okay, those are the red molecules of RNA are transcripts, right? That's mRNA. And you can see that even though in some it is not complete yet, okay, all these molecules are not complete yet. All these RNA molecules, the transcription isn't finished. As soon as RNA appears, ribosomes, those bell-looking shapes, bind the RNA molecule and start to produce protein. Does that make sense? Again, the analogy that I make for this, like reading a novel in some kind of a magazine. You know how they used to publish novels chapter by chapter in a magazine? So you get the first issue of the magazine, you read first two chapters. Okay. While you are reading that, the author writes the third and fourth and publish it in the second issue. While you're reading third and fourth, author writes the fifth and sixth. Does that make sense? So the process of transcription and translation in prokaryotes is continuous. Translation occurs on the incomplete transcript. The question that we have to ask ourselves, how is that possible? And more important, why it is possible only in prokaryotes and not eukaryotes. What's the fundamental structural difference between eukaryotic and prokaryotic cell? They anucleated. What does that mean from structural standpoint? Is where? Where's the DNA in prokaryotes? No, cytoplasm. Yeah, right. In prokaryotes, in the cytoplasm. Does that make sense? And in eukaryotes, where is DNA? In the nucleus. And nucleus is bound by what? Well, nuclear membrane, right? Nuclear envelope. So when DNA is transcribed into RNA, what does RNA have to do to serve as the template for protein synthesis? Leave the nucleus to go to where? Endoplasmic reticulum. So you have spatial separation. Does that make sense? Transcription happens in the nucleus. Translation happens in endoplasmic reticulum. And they are separated by the nuclear envelope. So there is no chance ribosomes get into the nucleus. Does that make sense? In eukaryotes. Alright? In prokaryotes, mRNA is produced, it hangs into the cytoplasm, and ribosomes just sit on it and start translating. Does that make sense? Another thing, there's one step of gene expression that is present in eukaryotes, but not in prokaryotes. Just discussed it. Splicing. Eukaryotic RNA has to be modified. Does that make sense? Does that make sense? It has to be edited. Prokaryotic RNA is ready to go. Does that make sense? So before we move on to ribosomes and genetic code, one of the big differences between gene expression in eukaryotes and prokaryotes, transcription and translation are practically simultaneous. They happen at the same time. They're continuous. In eukaryotes, they are separated in time because of the splicing, and in space because transcription happens in the nucleus and translation in the endoplasmic reticulum. Ribosomes. Those are not membrane-bound organelles. They consist of proteins and RNA, and they are significantly different. Prokaryotic are 70S. We discussed it, 30 and 50S subunits, two subunits, big, large subunit, and the small subunit. And eukaryotic ribosomes consist of also small and large subunit, 40 and 60S, but eukaryotic ribosomes are bigger. So you can see that there's a structural difference between prokaryotic and eukaryotic ribosome. Now I announce it's not so easy question. If you have bacterial ribosome and ribosome of a human that are very structurally different, what does it mean for healthcare? They have the same function, but they structurally different. What does that mean to healthcare? You have to make like a huge leap of logic. Not huge, huh? You're going in the right direction, but it's not about more. You're going in the direction of treatment, yes.
Based on their structure, you're going to have to treat each one in a specific way because of the structure.
Okay, so they have different structure. So when you say treatment,
like if it's a bacteria, for example, you would probably have to select which antibiotic it's going to respond to based on the structure.
Okay. Now, if they're structurally different, can you block bacterial ribosomes from working while leaving eukaryotic ribosomes intact? If they are structurally different. That's a simple question. You can. Does that make sense? If you have bacterial ribosomes different from eukaryotic ribosomes, you can design a drug that would shut down bacterial ribosomes, specifically targeted, and leave eukaryotic ones working. So you can selectively treat bacterial infection without very much affecting function of the human cell. Does that make sense? When we'll get to the antibacterial drugs, you will see that the majority, vast majority of the drugs that we use to treat bacterial infections, they target ribosomes, target protein synthesis. Examples are tetracyclines, erythromycin, clindamycin, oxazolidinones, neomycin, kanamycin, just huge number of different drugs. They all target the ribosome because it's such a great target, you know? They're so different. Does that make sense? Just to give an idea, mitochondria and chloroplasts obviously have 70S because they used to be bacteria, right? So before we move on to genetic code, what you have to understand about ribosomes, the importance for the pharmaceutical industry, for healthcare, importance of the difference between the bacterial and eukaryotic ribosomes, that we can specifically target one of them. And the structure, they have two subunits, small and large subunit, and they consist of proteins and ribosomal RNA. Does that make sense? Can we move on to the genetic code? So it turns out that all human proteins consist of, well, not only human, all proteins consist of 21 amino acids. On this picture, you see only 20, but I'll mention 21. So you probably, I don't know if when you took previous biology classes, were you told it's 20? 21? Yeah, so 21, these are 20 conventional amino acids here. There's also amino acid called selenocysteine. It's amino acid cysteine. Where the heck is this? Okay, cysteine, in which sulfur atom is substituted by selenium atom. Okay, it has its own codon to encode, so 21 amino acid. There were debates about 22nd, but they kind of died out. So 21 amino acid, each amino acid is encoded by the combination of 3 nucleotides. Not 2, not 4, but 3. Why 3? That's a little digression into the math. The question is, how many nucleotides you have? Different types of. How many nucleotides, not amino acids, how many nucleotides you have in the DNA? 4, right? So you need to produce at least 21 combination, okay, 21 different combination using 4 letters. So the question is, how many letters you have to use in each combination? What if you use 2? How many combinations are going to be there? You have 4 letters and you make 2-letter words. How many words? The answer is 16. It's 4 in the power of 2. The trick is very simple. Do you know code locks? That I will teach you a little math trick. The locks, right? That have like code dials. You know what I'm talking about. So imagine you have a lock with 3 dials. Each dial has numbers from 0 to 9. How many combinations can this lock have? Huh? What is going to be the first combination? All zeros. Well, okay, you can do all 9. You can dial down, but it's fine. But all zeros. Then you take 1 dial and switch it to 1, 2, 3, 4. When you reach 9, like 009, how many combinations you tried? All zeros? Huh? No. All zeros, then 1, 2, 3, 4, 9. You tried 10. Okay. So then you move to the second dial. So pretty much you start to count from 0 to 999. Does that make sense? Because the first combination, all zeros, the last combination is 999. All numbers from 0 to 999. How many combinations total? Huh? 999 plus 1, which is 0. 1,000 combinations. You know why 1,000? Each dial has 10 numbers. We have 3 places, 10 in the power of 3. Does that make sense? If you have 4 dials, 10 in the power of 4. I tried it myself, my brother. I wanted to open my brother's case. He had a combination of 548. It took me about 25 minutes to reach the combination. Played a prank on him. It was pretty bad. He didn't know I would be so stubborn to actually manually go from first to last. Well, not last. Anyway, so if you have 2 nucleotides, you're going to have 16 combinations. It's not enough. What if you have 3 nucleotide words. It's going to be 4 in the power of 3, right? This is going to be number 64. You have 64 different words, but you have only 21 amino acids that you need to encode, which means quite a few amino acids will have more than one 3-letter word that encodes them. Does that make sense? They're going to have synonyms. This feature of the genetic code, that each amino acid is encoded by more than one codon, is called redundancy of the genetic code. Sometimes it is also said that the genetic code is degenerate. Does that make sense? A lot of amino acids are encoded by more than one 3-nucleotide codon. Do you follow me? Okay, now why it's so cool? Because if there is a change in, let's say, third letter of the codon for serine, the amino acid that will be a result of that codon is not going to be changed in the protein. Does that make sense? Think about this for a second. The function is the protein. If you change amino acid in the protein, what's going to happen to its structure? It's going to change. What's going to happen to its function? It's going to change. Now, if you have some way to prevent this, some way to reduce the effect of mutation, okay, on the protein sequence. Imagine that you have DNA, and in DNA, A is being substituted by G. In RNA, therefore, U is going to be substituted by C. But it's fine. We still have the same amino acid. Do you see what I'm trying to say? It kind of reduces the effects of the mutations in the protein sequence. Okay. Now, when amino acids participate in the translation, how are they transferred to the ribosome? Transferred by the tRNA. Okay. And now, one thing that I want you to understand really clear is the concept of anticodon. Okay. Each transport RNA has anticodon loop. You can see it in the right bottom corner. Anticodon on amino acid is complementary to a specific codon in the mRNA. For instance, this amino acid, this one, has anticodon CGG, which is complementary to the codon GCC. Let's figure out what this codon encodes. GCC, here, GCC, is the codon for alanine. Make sense? So this tRNA will carry which amino acid? Alanine, yes? That makes sense. So you have, considering that you have 3 codons that serve as the stop for protein synthesis, you have 61 codon that carries information about amino acids. So you're going to have 61 different types of tRNA, each of them recognizing a particular codon. Before we move, that's very information dense, and we're going to do a little play here to figure out how protein synthesis happens. Okay? Before we move on, things that you have to understand. What is redundant or degenerate code? Okay? The concept of codon, that it's triplet, okay, consists of 3 nucleotides. Each 3 nucleotide encode amino acid or can serve as the stop for the protein synthesis. And the function of tRNA, that tRNA has an anticodon that allows it to recognize a specific sequence in the messenger RNA. Now the protein synthesis. In both eukaryotes and prokaryotes, protein synthesis starts when small subunit of ribosome recognizes a special sequence in the mRNA. In the prokaryotic one, the sequence is called Scheindel-Gardner sequence. In the eukaryotic, it's a special structure in the 5' end of the messenger RNA called 5' cap. So small subunit binds to this starter sequence. Okay? And first transport RNA binds to the start codon. Start codon always encodes methionine. There are no exceptions. The very first amino acid is always methionine. Does that make sense? Start codon is universal. It's AUG, and it encodes methionine. Okay? Finally, the large subunit joins the party, and you have fully assembled translation complex. You can see it here in the left bottom corner. So you have small subunit, which is blue, large subunit, which is beige, tRNA, dark blue, and purple mRNA. So everybody's in place, okay, and synthesis can start. The next step is the addition of tRNA to the A site of the ribosome. Ribosome has 3 sites: A site, B site, and E site. Okay. So next, tRNA enters the A site. This time, previous tRNA carries the growing polypeptide chain and sits in the P site. The entire polypeptide chain is being transferred to the incoming amino acid. Ribosome shifts again, and the chain goes from A to P site. It all sounds a lot of gibberish. Give me a second. We're going to do an actual representation of what is going on, and it will make it a ton clearer. Okay. The tRNA that has no amino acid anymore is being thrown out from the E site. This process repeats itself again, and again. New tRNA enters the A site. Often call it attachment. Polypeptide chain, growing chain, is moved from P to A. Ribosome shifts. Empty tRNA is thrown away. The cycle starts again. When does it stop? When the the termination factor, the release factor, binds to the stop codon, UAG, and makes polypeptide to detach from the ribosome. Selenocysteine amino acid is inserted when there's a special sequence after the UGA stop codon. This sequence, called sec insertion sequence, makes stop codon work as the codon for selenocysteine. This entire process requires energy and is pretty fast. In E. coli, insertion of one amino acid takes 5/100th of a second. So if you have a protein that consists of 100 amino acids, 5 seconds and the protein molecule is produced. Okay? Now, Is that the one? Yeah, I think so. So take home message. Understand the process. That's all I can say. Okay. So like, where does the tRNA with the new amino acid come in? A side. Where is tRNA with growing polypeptide chain? B side. Does that make sense? What I really find fascinating about this process, just a little rambling here, is that it's not new amino acid that's being added. It's the entire growing chain that's being transferred every time. Does that make sense? Okay. So this is the summary of, should I say, yeah, summary of differences between the eukaryotic and prokaryotic gene expression. In prokaryotes, as you can see, the process is fast and straightforward. Transcription and translation happen practically at the same time. They're almost simultaneous, they're continuous. Okay, so translation follows the transcription. mRNA does not require processing, and single mRNA transcript can contain more than one gene. This is called polycystronic mRNA. Does that make sense what I just said? One mRNA may have more than one gene. So many genes can be translated at once. Okay, and of course the ribosome is 70S. In eukaryotes, it's more complicated. So you have transcription that goes on in the nucleus. Modification of RNA, splicing and addition of some other structures also happens in the nucleus. And only after that, the modified, spliced, capped RNA is exported into the endoplasmic reticulum or the cytoplasm, where it is translated. You see the difference? And finally, in eukaryotes, mRNA is monocystronic, which means you have an mRNA molecule that always contains only one gene. Got it? So I'm gonna ask you the difference between two processes: the process in eukaryotes and process in prokaryotes. Okay, I'm getting the new version of study guide ready. Speaking of which, first of all, when I ask you, did you like something that I do? I'm not asking it to hear, oh yes. I'm asking it to know if there are certain things that I can do better. And believe me, there will be no retaliation. I would be, unless like you call me really bad words, like, or you offend my family. Okay, it's more about family insult than insult against me. So that study guide, was that convenient? Or you want, okay, good. Because when I write the exam, I actually look at the things that I write in the study guide. Okay, so I try to do as much similarities in questions as possible. Okay, so I'm going to keep doing the same type of study guides, and I'm getting ready for the second one. So once the protein is made, question?
This isn't, no, we're starting something new, right? Like,
Oh, that's not on the exam. No. That's the new material. That's not in the exam. Don't worry. And so I give you extra time to get ready for the exam. I prefer, and students generally prefer, to take exam after the weekend because it gives time. Although we start the new material, it gives time. Okay, we may not be able to do it always, but I mean, sometimes if we end up with if we finish material on Monday, I'm not going to wait for a week, okay? We're going to take it probably a couple days later, okay? But I try to give you some extra time to prepare. So, I will ask you about the differences in eukaryotic and prokaryotic gene expression. So understand these pictures, these tables. Proteins, mostly in eukaryotes, by vas dominance, can undergo so-called post-translational modification. You can add sugars to them, I don't know, acetyl groups, hydroxyl groups, carboxyl groups. If they are faulty, there will be so-called ubiquitinated, so the small protein called ubiquitin will target them for destruction. There are plenty of modifications. In which organelle do they happen, those modifications? Protein modification in eukaryotes. Protein is synthesized in rough endoplasmic reticulum and is modified in no. Smooth is lipid and carbohydrate metabolism. Some sort of a complex, Golgi. It's modified. Proteins are modified in Golgi. Does that make sense? Okay. So that's the summary of process. If you prefer flow charts to the text. That's a really nice summary here. That's simple prokaryotes. Okay? DNA, RNA, protein. That's what happens in the eukaryotes. Different regulatory sequences, exons, introns, splicing that results in different types of transcripts. Each transcript produces different proteins, and then proteins undergo post-translational modifications. Does that make sense? In terms of the studying approaches, I told you before to tell the story, to talk to somebody. You don't even have a partner for things like that. You can try and close everything that you have, PowerPoints, books, whatever, and try to draw the flow chart by yourself. Are there going to be parts missing? Probably so. But you will be able to identify what are the weaknesses. Where do you have a weakness? Does that make sense? Come back and ask, okay, I don't really understand this. I don't really get this. I don't remember this. Why is that so? So we'll be able to question it. And if you cannot find the answer, email me. Right? I'm checking my email pretty regularly, so you can expect a pretty fast answer. We got it? Replication. Replication is copying of DNA to pass it on to the next generation. Before we move on, does replication have to do anything with expression of genes? No. Those processes are not intertwined. They are not essentially connected. Does that make sense to you? When we replicate DNA, we do not produce any proteins. We just make a copy. Like when you're copying a document, it doesn't mean that you read it. When you use the copy machine, You don't read the document. You don't have to. You just can copy, right? So that's an important moment to remember. DNA replication has nothing to do with the gene expression. So what are the steps? First of all, the helix of the DNA is getting unzipped. Okay. And then you have by the enzyme called helicase. And then you have enzymes called DNA polymerase. DNA polymerase makes copies of each strand. Okay. And you used to have one molecule of DNA, now you have two. Does that make sense? Okay. Why DNA is double-stranded anyway? I mean, you can take away one strand, you're still going to have all the same information. From the standpoint of survival, why do you need two complementary strands of DNA? Huh? Physically, chemically, we don't really know. Well, it's hard to judge because it's hard to say because we don't know what was the selective pressure, billions of years ago. DNA carries the genetic information. The sequence of DNA, does it have to be preserved? What if DNA gets like smashed, squished, there breaks in the DNA? Is that bad? Yes, that's bad. If you have one strand, yeah, if you have one strand and and something is messed up in that strand, that's it. Cell may die. If you have two strands, one of them gets messed up. The mess up can be repaired. Does that make sense? Because you have a complementary sequence. Huh? You have a blueprint to follow. Very good, yes. Does that make sense? Now, what if you have double strand mess up? Oh, you boned. That's a big problem. Like gamma radiation can break both strands at the same time. That's really, really bad. We have some mechanisms that said they're not, 100% reliable. So that's why we have two strands, to protect against possible alterations in one of them. Right? Replication is pretty conservative, which means it is very similar in prokaryotes and eukaryotes. Okay? And obviously in eukaryotes, replication happens in the nucleus. Does that make sense so far? Now think about this. DNA, as it turns out, is always confined to the nucleus. Right? It never leaves the nucleus normally. It is replicated in the nucleus. It is transcribed in the nucleus. What if DNA in the cytoplasm? What does that mean? Something's not right, something's wrong, yeah. And there's actually special receptor systems in the cytoplasm that detect DNA in the cytoplasm. And usually they kill the cell if there is a DNA in the cytoplasm. As the protective measure. So again, replication seems fairly simple in that picture. The process involves many proteins, okay? And I made a list, table, the name and the function, and kind of giving you heads up, you have to recognize. So you may encounter questions in the next exam. Description of the function, you have to pick the right enzyme. Or name of the enzyme and you have to pick the right function. So there are two enzymes called DNA polymerase. DNA polymerase 1 and 3. DNA polymerase 3 is the main copy machine. it produces a new strand using old one as the template. Okay. DNA polymerase 1 removes so-called primers, RNA primers, and replaces the former RNA with a new DNA strand. Now, those primers, short fragments of RNA that are necessary to start replication, are produced by the enzyme called primase. Okay. There are some gaps in the freshly synthesized DNA molecule. To fill those gaps, enzyme ligase, actually seal the gaps, okay? That enzyme called ligase stitches fragments that have little gaps in between them. If you look at this picture, you will see that double-stranded DNA molecule has to be unzipped. Unzipping is performed by the enzyme called helicase. When two strands of DNA are unzipped, what do they try to do? Go back together. A protein called single-strand binding protein, SSBP, prevents them from going back together. The special protein called, I like the name, sliding clamp, holds DNA polymerase in place, not letting it fall off the replication fork. Finally, think about this. You have a double-strand DNA, which is shaped like a helix. Did anyone use the landline? You know, the phone that's actually connected to the land. Okay? And not the phone that you can go with through the house talking, but actually the phone that is connected by this spiral wire to box. Do you know those? You know those? What happens to that spiral wire? It gets all, it can stretch, but if you, like, you know, if you walk around and just, tangles. It gets all coiled up, right, and tangles. Same happens with the DNA. There are two enzymes called topoisomerase 2 and topoisomerase 4 that untangle. Topoisomerase 2, or DNA gyrase, straightens up DNA molecule so helicase can unzip it. And topoisomerase 4, you will see how it happens. It separates two newly synthesized double-stranded DNAs in bacteria, because it turns out bacterial DNA after the replication will look like this, like two circles concatenated with each other. Okay, we'll get to that. We're gonna unveil what's going on during replication. What did I want to say? Okay, I'll figure it out, what I wanted to say. This is the typical replication fork. There's a mistake in that picture. Can you find it? You know about that. I don't have to tell you anything. You know that. There's a mistake on that picture. Exactly. Can you see that? There's 5' and 3' on the left, and they are arranged in the same way on the right, which is wrong. Does that make sense? So that's bad. Okay. So DNA polymerase reads the DNA from 3' end towards 5'. should be 3, this should be 5. Does that make sense? Okay, so far so good. So now think about this. If you have initial DNA molecule, you get the DNA unzipped, and each old strain, which is shown here in blue, is copied, complementary, and this copy is the new strand, which is shown here in red. So your two new molecules consist of one old and one new DNA molecule. Does that make sense? This is called semi-conservative replication. Semi-conservative. That each new molecule of double-stranded DNA consists of one old strand and one new strand. Replication starts at the origin of replication, which is reach in the AT pairs. Why? Weaker bond, it's easier to unzip. Okay? And at each origin, two forks, replication forks are formed. So this is one fork, this is another fork. And they move in opposite directions, like two zippers. You know, if you put them in the center like this, things with two running thingies, and you start to unzip in opposite directions. Make sense? Okay. So eukaryotic DNA has usually multiple origins of replication, as demonstrated here. And these multiple origins of replication allow to copy the DNA in highly efficient fashion. Well, think about this. You have 3 billion nucleotides. To make a copy of 3 billion nucleotides, you have to be efficient. You have to have multiple origins of replication. Like if you need to copy 1,000 pages, you probably wouldn't use just one copy machine. You would ask 10 people, and they would copy it in 10 different copy machines. Does that make sense? Bacterial chromosome has one origin of replication. Okay? So this shows you the origin of replication, for instance, in the bacterial chromosome. So now the process. Step by step. Okay? Imagine you have double-stranded DNA, it's helix, and it's all tangled up. All tangled up. What do you have to do first? Detangle it, straighten it up. That's topoisomerase 2. Untangles. It's still double strand. So what do you have to do with the double strand then? Unzip it. It's a helicase. Right? Once you separate two strands, what do they want to do immediately? Yes, they want to come back together, and single-stranded binding protein prevents them from doing so. Okay? And finally, your DNA polymerase 3 can start making DNA of the new strand, synthesis of the new strand. There's one little problem. In order to start the replication, DNA polymerase cannot just, sit and start the replication. It needs what? RNA primer. It needs RNA primer. That makes sense? Just so it happens. It's just the way it works. It's an RNA primer. So the enzyme called primase, which is shown here as the green box, produces the RNA primer. And then DNA polymerase starts to synthesize DNA, reading the original, the old strand from 3' to 5' end. Does that make sense? Now, this fork moves in which direction? To your left or to your right? It unzips. In which direction it moves? Huh? To the left. Right? Because this is where it started. Does that make sense? Do you understand that? This is where it started initially. It unzips and starts to make new strands. That's where the new strand started from, on the right. And the fork on this picture moves to the left. And if you would look at this strand that I just painted black, okay, it's not a problem to read it from 3' to 5', because so it happens, it also goes to the left, from right to left. Does that make sense? Now, let's take a glance at what's going on the opposite strand here. So on this strand, if you will try to read it from right to left, it's going to be from 5' to 3'. Can DNA polymerase do that? Can it read an opposite? It cannot. So you have to, develop the way to overcome that. And that is replication when DNA polymerase, now look at this, just imagine how it goes. You have DNA polymerase, where it is, DNA polymerase, that produces a short fragment called Okazaki fragment. It's a start of synthesis here. The red ones are Okazaki fragments. DNA polymerase in this case reads from 3 to 5'. Produces Okazaki fragments, then it jumps back and will make another Okazaki fragment. Then it will jump back and make another Okazaki fragment. Does it make sense? Yes? Oh, no. It goes... let me pause it for a jump. So, sorry, this strand is called leading. This strand is called lagging. So DNA polymerase 3 performs a continuous, uninterrupted synthesis of the new DNA strand on the leading strand. And on the lagging strand, DNA polymerase 3 makes discontinued synthesis of Okazaki fragments, okay, from which primer is removed and primers are stitched together by the DNA ligase. Does that make sense? But if I will try to practice my jumps for the next semester. Okay, I can jump really far. So what happens essentially on the leading strand? You have one long uninterrupted new strand. On the lagging, you have short fragments that eventually are going to be stitched and you won't be able to tell any difference. Does that make sense? Sorry. Okay. Now, in bacteria, you have single circular chromosome. When the entire chromosome is copied, two copies become tangled like this. To separate them, topoisomerase 4 introduces the break in one and then stitches it back together. And now you have two separate chromosomes. Does that make sense? Replication's over. It's done. Before we move on to mutations, again, functions of the enzymes. You have a table. That's your reference. That is your reference. You won't have to write me a list, but you will have to answer questions. The enzyme that stitches Okazaki fragments on the lagging strand is DNA ligase. Does that make sense? Or the function of helicase is to unzip the strands of the DNA. We good? Is that understood? Awesome. You have to understand what is conservative, what is semi-conservative replication is. You have to understand what is origin of replication. All you have to do, speaking of understanding, it's the point where replication starts. Why it is AT reach? It's easier to separate. All right? Understand the sequence of events in the replication. There's something else. Oh, what is Okazaki fragments? On which strand are they formed? The lagging. What is the difference between lagging and leading? Lagging is interrupted synthesis. Leading is continuous synthesis. Okay, that's basically it. All right. Let's move on to the mutations. I don't know. Did you ever see in the media, like when they write about science, diseases, they say mutant protein? Well, if you ever see that, mutant protein, that's wrong. And I'm going to be the first who will cast the stone towards myself. I sometimes say it, but strictly speaking, it is a wrong expression. Mutation can occur only in the nucleic acid. That's the definition of a mutation. It's the change of nucleotide in the nucleic acid. That change of the nucleotide can lead to changes in the protein. That is right. Does that make sense? So, mutations can be of different types. We're going to be talking mostly about the point mutations. Point mutation means that only one nucleotide is substituted, inserted, or deleted. Right? So, mutation is considered to be silent when substitution, substitute A to T, okay, when substitution doesn't change the amino acid in the protein. Does that make sense? Because both the GTA and GTT in the DNA, they both encode for valine. Okay? Silent mutations, no effect on the sequence. What if substitution say cytosine is substituted by adenine, CCC was codon for proline, ACC now a codon for amino acid threonine. This mutation is called missense. It's a new, different amino acid. Instead of proline, it's now threonine. What effect is it going to have on the protein? It's going to change what? First of all, if you change amino acid, it changes structure, meaning changing. If it's structure, it changes function, right? And changes phenotype. You have to understand a few things. Let's be clear on the mutations, because there's a lot of misconception about this. First of all, sometimes when you substitute one amino acid, nothing happens. Okay? Although amino acid is different, it may not have a profound effect on the protein. Is that clear? Second, if the protein is absolutely necessary, if it's like very, very important protein, and its function is completely screwed up, and it doesn't work anymore, what can be the ultimate outcome of this for the cell? Death. Some mutations may not be like compatible with survival, okay? You can, let me put it this way a little bit, again, digression in science. We now can manipulate with animals, okay? We can alter genes in mice. And what is the best way to study what a certain gene does? To change it, to kill it, okay? To destroy its function. You know? Like, you don't know what is the role for transmission in your car until transmission is completely done, okay? So, same goes for the genes. Loss of function. You destroy the gene and then you look at the phenotype. Let me tell you something. The biggest bummer is when you invest money and work and time in knocking out a certain gene in mouse, and you try to produce the offspring. There's no offspring because it's embryonically lethal. That happens sometimes. So embryos don't even, like, they form and they die. They form and they die. So you don't even know what it does. You only know that it's really important. That kind of sucks often. So that mutation can be absolutely unnoticed in terms of effect on the protein, or it can lead to such changes in the protein that, you know, everything is screwed up. Somewhere in the middle, the great example of it is, of course, sickle cell anemia. Okay, it's the classic example of missense mutation. One nucleotide substitution leads to substitution of one amino acid. I don't remember which one to which. I remember that valine is involved. So you have one amino acid difference in one chain of hemoglobin, which leads to completely dysfunctional red blood cells. Okay. What can also happen if you, let's say, if you have amino acid that is required for the protein modification, if amino acid is phosphorylated, you change amino acid, there's no phosphorylation, there's completely different signaling in the cell that may lead to various effects. So that's the missense. Nonsense is when a codon for amino acid, like this TAC, is substituted for the stop codon, TAG. So what's going to happen if somewhere in the middle of the protein, somewhere in the middle of the gene, you will introduce the stop codon? Which effect is going to have on the protein synthesis? Oh yeah, it's just not going to be complete, right? It's going to be devastating, right? So often these proteins are called truncated. cut in half. Usually nonsense mutations lead to complete destruction of the function, loss of function. Does that make sense? We're good in that? So again, before we move on to other types, silent, no effect on the protein synthesis. Missense, new amino acid instead of the old one, and nonsense, stop codon instead of amino acid. What if you will insert or remove a nucleotide? Like here, you have TA, you remove a couple of nucleotides. What's going to happen to the amino acid sequence? Huh? It's going to change in everything after the insertion and deletion is going to change. And let me try to explain you why. When there is a translation, each tRNA using its anticodon loop recognizes what? Anticodon loop recognizes codon. Does that make sense? Anticodon recognizes codon. Right? Now, if you remove 2, then the next codon will start from the wrong nucleotide. Does that make sense? Essentially, translation goes, so mRNA is read in frames. You read by 3 letters. Does that make sense? First 3, second 3, third 3. If you remove these 2, your first codon is going to be the same, AGC. But second one is going to be totally different because your entire sequence will shift. Does that make sense? The entire sequence will shift. And the frame of reading, so-called reading frame, will shift. This mutations, insertion or deletion of 1 or 2 nucleotides, is called a frame shift mutation. Does that make sense? It is shifting the frame. Okay. Example of the mutagen, example of the agent that can do mutation is the nitrous acid, HNO2. It changes cytosine to uracil, okay, and as the result, you have a different pair. Does that make sense? Now, before we will talk a little bit more about nitrous acid and why this example is quite relevant, summarize what you have to know. If I give you a definition of the mutation, okay, insertion of 1 nucleotide into the sequence, into the DNA, will lead to frame shift. Mutation that doesn't affect the protein sequence is silent. Mutation that introduces the stop codon is nonsense. Again, as I mentioned, you have to match name and the definition. Does that make sense? Now, what is the importance of nitrous acid? Go to chemistry, inorganic. What's the name for NO2 anion? What do we call this anion? It's a nitrite. Doesn't ring the bell? Who likes bacon? Cured bacon? Anybody? Okay. Next time you buy cured bacon, no, seriously. Next time you buy cured bacon, any type of cured meat, bacon, sausage, read the composition. There's nitrite. Nitrite retains the red color. Okay. It's a mutagen. And recently, a study was published in Britain that actually linked the consumption of processed meat, specifically processed meat, to an increased risk of colon cancer. Of course, it doesn't mean that if you eat bacon, you're going to die of the colon cancer. You have high risk of that. They established sort of a safe level, which is laughable. It's like 2 slices of bacon. You gotta be kidding me. And I cannot survive on 2 slices of bacon a day. There is a solution, by the way. Now, I know it's not microbiology, but seriously, they sell uncured bacon now. Taste-wise, it's perfect. Just no nitrite. Okay? So, just an idea of what you get exposed to when you buy stuff in the store. So, what can cause mutations? Agents that are called intercalating agents. So what they do, agents like acridin, they incorporate themselves between the base pairs in the double strand. So imagine I have two pairs, bless you, 2 pairs, those are my palms, and something gets incorporated in between them. Okay, 2 pairs, something gets incorporated between them. It causes the disturbance in the DNA structure. Okay? And when the DNA structure is replicated, okay, when there is a replication, there's going to be a deletion or insertion. There's going to be some sort of maybe frame shift, insertion of additional amino acid. There's going to be something, okay? When DNA structure is changed, it is not replicated properly. Examples of such agents are ethidium bromide and acridin orange. You're not going to find them in the regular life. That's a lab reagents. Ethidium bromide and acridin orange are used to detect DNA actually, because when they intercalate, when they stick themselves into the DNA molecule, they make it more visible when you shine the UV light on it. I was told that they are so carcinogenic. If you put a drop of ethidium bromide on the skin, you're going to have skin cancer. Not melanoma, but you're going to have skin cancer. Never tried, always used gloves. Worked with both so far. Don't want to brag, but nothing yet. I don't know, maybe in the future. Okay? So, they are extremely carcinogenic. Ionizing radiation. It introduces the break repairs. Repairs may be single strand, which is not a big deal, right? Or double strand break. Okay? If the break is double strand, then the repair system may not correctly repair, and that may cause mutations, alterations to the sequence.
Radiation poisoning or sickness is pretty nasty.
Yeah. Oh yeah, and that's actually, it's a double strand breaks in the DNA of the cells that divide, especially they affect cells that divide frequently. For instance, cells of your intestinal lining, of your stomach lining, they changed every week or 2. Skin the same, yes. Okay, and cells that divide frequently, they're affected the most. And that sucks. Non-ionizing radiation, like a UV, it doesn't break the strands, but what it does, it forms the pyrimidine dimers, two adjacent residues of, let's say, thymine, okay? They link to each other, and obviously, when these guys are linked, there's going to be a misreading during the replication, okay? Now, before we finish, a few things. What you have to, so, mechanism of intercalating agents. What is intercalation? Insertion in between the pairs. What does gamma radiation do? Single and double strand breaks. What does UV light do? Pyrimidine dimers. Before I finally let you go, quick question. I used to have a well, she's still alive, I hope. I had a colleague. She studied double strand break repair of that stuff. Very fundamental, like theoretic, well, very fundamental features of the cell. She had pretty good funding from one government agency, which quite a few employees of which are exposed to ionizing radiation on the regular basis. Any idea what the government agency? Nope. First thing, but no. Guys that work with nuclear stuff, they're actually well protected. Those guys cannot be well protected. NASA. Astronauts in space are exposed to all kinds of cosmic rays, which are high energy. And she was studying the mechanism to help understand how risky it is for them in terms of mutations. She had a pretty good money from them.